Hi everybody, we are learning question 27 from GATE Biotechnology 2022 question paper carrying 1 mark. The question is, assuming independent assortment and no recombination, the number of different combinations of maternal and paternal chromosomes in gametes of an organism with a diploid number of 12 is. So we are now required to calculate the number of possible combinations in which the two parental chromosomes combine to form an offspring gamete. In sexually reproducing organisms, individuals inherit a set of chromosomes from each parent. These chromosomes come in homologous pairs, with one chromosome in the pair coming from the mother and the other from the father. Each pair contains two different alleles of the same gene. So, independent assortment means that the maternal and paternal chromosomes in each pair can segregate into different gametes in various combinations. And recombination is when sections of homologous chromosomes exchange genetic material, but in our case, there is no such recombination. So, the number of different combinations of maternal and paternal chromosomes in gametes is given by the formula 2 raised to the power n, where n is the number of homologous chromosome pairs. The formula arises from considering how many different combinations of maternal paternal chromosomes can be formed in gametes. For each homologous chromosome pair, there are two possibilities for how the parental chromosomes can segregate into gametes. This is because during meiosis, each chromosome in a pair can go into either the sperm or the egg with equal probability. So if there are n homologous chromosome pairs, then the total number of different combinations of maternal and paternal chromosomes in the gametes is calculated by multiplying the possibilities of each pair. Therefore, the formula is 2 raised to the power n. So in our question, it's given that the diploid number is 12. So that is 2n is equal to 12. So there are 6 homologous chromosome pairs. That is n is equal to 6. So each pair consists of one chromosome from the mother and one from the father. Hence, the number of different combinations 2 raised to the power n is equal to 2 raised to the power 6, which is equal to 64. Therefore, there are 64 different combinations of maternal and paternal chromosomes in the gametes of an organism with a diploid number of 12, assuming independent assortment and no recombination. So the answer to this question is 64. So given that we have solved the question, let us understand more about some terminologies we saw in the question and the laws of inheritance. So to start with, genetics is the scientific study of genes and heredity, of how certain qualities or traits are passed from parents to offspring as a result of changes in DNA sequence. A gene is a segment of DNA that contains instructions for building one or more molecules that help the body work. So the common terminologies used in genetics are as follows. So one is trait, which is the morphological, anatomical, or behavioral feature of an organism. And allele. So allele is one form of the gene, that is, a specific composition of nucleotides would render a yellow seed color, while another would produce a green seed. This is just an example of how the same gene can give rise to different phenotypes, and that is called an allele. And an individual may have two copies of the same allele in the diploid genome. In such case, they are called homozygous. That is, in both the copies of the chromosomes, they have the same allele. So those individuals are called homozygous individuals. And an individual could also have one copy of each of the alleles, and they are called heterozygous. And phenotype is a term used interchangeably with trait. So it means the observable characteristic of an organism, like height, that's like tall or short, and skin color, and so on. And genotype is the genetic composition that determines a specific phenotype. For example, tall individuals would have two alleles, T and T, and short individuals would have small t and small t, and heterozygous individuals would have one allele for the tall characteristic and one allele for the short phenotype. Although, since tall is the dominant trait, the individual would be tall. So the underlying nucleotide composition that gives rise to the phenotype is called the genotype. So here are some more terminologies. So F1 generation is the first generation of offsprings as a result of mating. And F2 generation is the second generation produced by crossing or by interbreeding within the F1 generation. And monohybrid cross is a cross or mating between two parents that differ only by one trait. And dihybrid cross is when two traits differ between the parents. So let's look further to understand these terminologies better with examples. So, like you all must have known, Gregor Johann Mendel was a German Czech biologist, meteorologist, and a mathematician. He was born in a German speaking family in the part of the Austrian Empire, which is today's Czech Republic, and he gained recognition as the founder of the modern science of genetics. So, although farmers had known for many ages that crossbreeding of animals and plants could favor certain desirable traits or characteristics, Mendel's pea plant experiments conducted between the years 1856 and 1863 established many of the rules of heredity, and now these are referred to as the laws of Mendelian inheritance. So, Mendel is called the father of genetics. So, Mendel worked with 7 characteristics of pea plants. So, plant height, pod shape and color, seed shape and color, and flower position and color. Taking seed color as an example, Mendel showed that when a true breeding yellow pea and a true breeding green pea were crossbred, their offspring always produced yellow seeds. However, in the next generation, the green peas reappeared at a ratio of 1 green to 3 yellow. So, to explain this phenomenon, Mendel coined the terms recessive and dominant in reference to certain traits. In this example, the green trait, which seemed to have disappeared in the first filial generation, is recessive and the yellow is dominant. So, dominant traits are always expressed when the connected allele is dominant, even if only one copy of the dominant trait exists. And recessive alleles are expressed only if both the connected alleles are recessive. If one of the alleles is dominant, then the associated characteristics is less likely to manifest. So Mendel published his work later in 1866, demonstrating the actions of invisible factors contributing to these characteristic traits, and now they are known and they're called as genes. So in predictably determining the traits of an organism. So the profound significance of Mendel's work was not recognized until the 20th century, so which was more than 3 decades later, with the discovery of his laws. So several other scientists independently verified several of Mendel's experimental findings in 1900s, and thereby it laid the foundation of modern age genetics. Mendel's experiments laid foundation to the laws of inheritance. One is the law of dominance. We just learned that every gene has 2 alleles for a specific trait or phenotypic characteristic. One allele is dominant and is always expressed, while the other allele is recessive and its characteristics will be suppressed by the presence of the dominant allele. For example, in Mendel's experiments, pure tall and short pea plants were crossbred. So one plant is homozygous tall, where both the alleles that determine the height of the plant encode the dominant trait, which is tall, and it's represented by capital T. And similarly, the other parent is homozygous short, where both the alleles correspond to a short height. So during meiosis, the homologous pairs segregate to form 2 gametes from each parent, all with 50% probability of being passed on to the offspring. So we could see that in the F1 generation, all the 4 offsprings are heterozygous, and since one dominant allele is expressed in all the offsprings, all of them were tall. So this proved that the dominant trait will always be expressed even if it is encoded in just one copy. Further, let's now learn about the law of segregation. So during gamete formation, Every paired alleles separate, that is, segregate randomly so that each gamete receives one allele or the other. So, two alleles of a gene present in the F1 generation do not mix with each other. They separate and pass on to different gametes in their original form, producing two different types of gametes in equal proportions. So the pair of alleles of each parent separate, and only one allele passes from each parent onto the offspring. So which allele in a parent's pair of alleles is inherited is a matter of chance, and they all have equal probabilities. So this is the law of segregation. Here's an example that demonstrates the law of segregation with Mendel's experiment in crossing tall and dwarf plants. So like we saw earlier, in the F1 generation, all the plants were tall but have gametes, that is the alleles for both tall and dwarf. So when progenies of the F1 generation are crossed within themselves, the second generation produces traits as follows. So we have two parents, both of them are heterozygous, that is one tall allele and one short allele. So when we cross them, we get one homozygous tall and one homozygous short plant and two heterozygous tall genotypes. So from this we could see that three of the offsprings express at least one tall allele. So we get three tall plants and one short plant. So in the second generation, we get traits in the ratio of 3:1. That is 75% of the dominant allele and 25% of the recessive allele. So we get three tall plants and one short plant. Further demonstration of the law of segregation can be seen when the F1 plants, that is, the heterozygous tall plants, capital T and small t, is crossed back to the parent dwarf variety with two small t's. We could see that it produces two heterozygous offsprings which are tall and two homozygous recessive which are short offsprings. So the phenotypic ratio is observed to be 1:1. This shows how the gametes from the parents segregate and determine the characteristics of the offspring. The final law of inheritance is the law of independent assortment. It states that genes for one trait are not inherited together with another trait. That is, the traits are transmitted to the offsprings independently of one another. This was demonstrated by Mendel's dihybrid cross, where two traits such as the seed shape and seed color were observed. It was seen that the segregation of the seed color was independent of the seed shape. Both parental and new combinations of the characters appeared in the second generation, that is the F2 offsprings. This showed that assortment of gene of a pair is independent of the other pair. Let's look at an example of the mentioned dihybrid cross to understand independent assortment where two traits are observed. So let's consider two parents, one with yellow seed color and round seed shape represented by capital Y and capital R, and another parent with green seed color and wrinkled seed shape represented by small y and small r respectively. Since these are pure parents, they have homozygous copies of each of the alleles. So it was seen that all peas were yellow and round in the F1 generation. Then, when F1 were self-fertilized, 4 phenotypes were observed. So those 4 phenotypes that were observed are yellow round, yellow wrinkled, green round, and green wrinkled. Let us now look closer into the genotypes to understand the phenotypes and the ratios observed. If you carefully look at this Punnett square table, you can see different combinations of the genotypes. So we could see one pure yellow round with two capital Y's and two capital R's, and two pure yellow hybrid rounds with two capital Y's, one capital R and a small r, and one pure yellow pure wrinkled, that is two capital Y's and two small r's, which you can find over here, and similarly as follows. So these different combinations of genotypes give rise to 9 yellow round plants, that 1, 2, 3, 4, 5, 6, 7, 8, and 9 yellow round plants, and 3, 1, 2, and 3 yellow wrinkled plants, and 3 green and round plants, and 1 green and wrinkled plant. So the phenotypic ratio in the F2 generation is 9 is to 3 is to 3 is to 1, and the genotypic ratio is 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1, which I understand is quite complex, but you could easily make it out by drawing a Punnett square and looking closely at the genotypes. So thus one trait does not depend on the other trait expressed, hence proving that the gametes are independently assorted to give rise to each observable trait. So, we have understood the laws of inheritance. So, these are because they demonstrated the pattern of inheritance of the character from parent to the offsprings. The law of independent assortment has laid foundation of inheritance, which claimed that different genes assorted themselves independently of one another during gamete formation and had established the relationship between dominant and recessive gene type. It also provided the reason for variation in offspring transferred from the parents to the offspring in the form of gene present in the chromosome of the gamete. So, looking back at our question of interest, hope you understand the terminologies used. Since there is no recombination and the gametes are independently assorted as a whole, the formula is straightforward. If recombination, that is shuffling of the independently assorted chromosomes, were to occur, it would be a complex problem with a different formula depending on the number of recombinations that have occurred. So, in the given question, the number of different combinations is just a product of the total number of possible combinations, that is 2 raised to the power 6, and hence 64 is the answer.